Hi everyone, thanks for the introduction. We are excited to share our recent advances at the Eurofins discovery using high-throughput spectral shift. Before we dive into today's topic, I would like to take a moment to introduce you Eurofins discovery. With our global footprint, we offer fully integrated drug discovery solution from hit finding, chemistry, to translational biology and safety pharmacology. So today I will explore how high-throughput spectral shift is reshaping hit finding strategy and accelerating the identification of novel modulators and degraders. So high-throughput spectral shift is a great addition to our hit-finding toolbox. We use it alongside other strategies such as activity-based screening, fragment-based approaches, and even AI virtual screening. To do that, we've got access to diverse libraries such as the Platinum Div with our 500 compound and the fragment prop set as well. And if needed, we can also screen external libraries depending on the project's needs. All the data we generate go through Genedata Screener, which helps us to analyze large data sets. Finally, this work is supported by a team with a strong expertise in biophysics, assay development, and high-throughput screening. So now let's look at how high-throughput spectral shift is applied for drug discovery program. It's a versatile platform that supports a wide range of affinity-based screening projects. So on this slide, you have an overview about the key deliverable. So first, high-throughput spectral shift is enabled to rapidly identify binders, and even at early stage, we can distinguish between allosteric and outperforms binding sites. It is also compatible for studying proximity-induced interaction with the identification of molecular glues. And finally, by using diverse and multiple screening conditions and different protein constructs, we can discriminate binding domain of interest or assess target selectivity during the screening phase. Before we get into the spectral shift, how it works, let's step back and compare it with other screening strategies available on this field. So here you have a summary about 4 screening technologies used for the characterization and the screening of small molecule protein interaction or to inhibit protein interaction. We have the high-throughput spectral shift that is the topic of today, but as well we have the affinity selection mass spectrometry or SMS, DEL for DNA encoded libraries, and HT-SpS for homogeneous time-resolved fluorescence. I would like to highlight the advantage of this high-throughput spectral shift that is shown on this first column. So it requires a very little protein amount, so you need 1 mix to do a screen, and we work with tagged or untagged protein in solution. It supports any compound libraries in 1 compound per well format and provide direct binding measurement, including KD value for binary and ternary complex. So unlike SMS or DEL, high-throughput spectral shift does not require any compound multiplexing or data deconvolution. You do not need any DNA encoded library to perform your screen. In terms of timelines, high-throughput spectral shift delivers quantitative and qualitative data in less than 2.5 months. So based on this comparison, we believe that this affinity-based approach using high-throughput spectral shift accelerate hit-finding campaign in our drug discovery project. So now I will hand it over to Maud who will explain the principle of spectral shift.
Thanks, Vanessa. Now let me quickly walk you through the key principle of the spectral shift technology developed by NanoTemper. After that, I will outline the general workflow we apply at Eurofins during our screening campaigns. And finally, I'll show you how the raw data are integrated and analyzed using the screener software from Genedata. Spectral shift is a powerful fluorescence-based biophysical technique to quantify the strength of molecular interactions. It relies on detecting changes in emission spectra of a fluorescently labeled protein, specifically at two wavelengths, 650 nanometer and 670 nanometer. The fluorophore environments play a key role in the spectral shift. It can be influenced by the ligand proximity, which can induce subtle spectral shift, conformational changes in the protein structure, and surface properties such as hydrophoby and charge distribution. Spectral shift analysis enables the characterization of molecular interaction in single-dose experiments. So a large library of compounds can be screened to detect binder from non-binder, and then binding confirmation can be performed using dose-response experiment, allowing the affinity constant determination, KD, by plotting the ligand concentration versus the ratio. Several labeling strategies can be applied depending to the protein construct used and its structural properties. For protein without tag, we can use covalent labeling consisting in attaching fluorescence dye to either accessible lysine or cysteine. Alternatively, we often rely on affinity-based labeling strategy using the his tag or bioting with a specific labeling site. What we like here is that the same tag is used for protein production and the labeling, and there is no additional purification step after labeling. Then the well scan can be used to check the quality of the labeled protein and identify individual quality issue. With the X-scan, we can quickly spot issues like protein clumping, dust contamination, and or missing fluorescence signal. Now that we review the basic of the assay principle, let me walk you through the typical workflow on the next slide. So how do we integrate this technology in our screening process? Once we have selected the compound library, whether it's internal or external, we follow a workflow that is designed specifically for the project. We use different robotics to handle the liquids, and for most of our assays, we rely on the eco-distribution for nanoliter dispensing into NanoTemper's 1536 well plates. During the assay development, we choose the best liquid handler to make sure the data is consistent and reliable. Then we read the plate using the Dianthus uHTS, which is super fast. It reads a full plate in under 8 minutes. And that means we can complete large screen in less than 2 weeks. And to manage all the data, we use a Genedata Screener, which helps us to keep everything organized and ensure high data quality. So now let's take a look at how we handle and analyze these large data sets. We use the Genedata Screener to manage the raw data to the final result. So from the left to the right, we first start by checking the quality of the run using metrics like Z prime or RZ prime factor. And Genedata also help us to correct any patterns or inconsistency we might see. Then we move on to the processing data. We apply fitting models to extract KD, key value like KDs. And from there, we can identify and classify the hits, whether they are autofluorescent, non-binder, or potential binders. And finally, all the data is integrated and reported in a way that it's easy to share and discuss. So to show you how these tools and workflow come together in a real-world project, let's dive into a case study on PIM3 kinase. So PIM3 is a challenging oncology target. We applied HT spectral shift alongside traditional HT-SpS and fragment-based approaches to identify novel binders. PIM3 is a promising drug target due to its role in promoting cell survival and resistance to apoptosis. However, in its structural similarity to other kinases makes selective targeting a challenge. So to address this, we explored these three complementary approaches. The first is the traditional HT-S screening cascade using the biochemical assay like ADP-GLO. The second is the fragment-based approach that uses biophysical method to explore new starting point for the development of novel molecules. But today we'll focus on this third strategy, our affinity-based HT spectral shift approach, which allows us to detect binding events with high sensitivity and high throughput. So we initiated this assay using a high-quality PIM3 protein that was produced at our protein production facility, Eurofins Discoverex. So the protein purity exceeds 95%, as shown by this SDS page GL. And to ensure its stability, we perform a nano-DSF analysis, which revealed an apparent melting temperature of 44 degrees, indicating good thermal stability. And before launching the screening campaign, we ran essential controls to verify that fluorescent labeling did not compromise the protein functionality. For assay validation, we use AZ1208, a potent and selective pan-PIM kinase inhibitor as a reference compound. We conducted a time course dose response analysis of AZ1208 at various incubation time. And the data here clearly show that the optimal assay window is achieved after 1 hour of incubation. This bar chart summarizes the key assay quality parameters that we follow over the time. The signal-to-noise ratio in blue, the delta ratio, and the KD values. After 1 hour, we observe an excellent signal-to-noise ratio and a stable data ratio maintained for up to 24 hours. And notably, the KD value confirmed the strong binding of the reference compound. So PIM3 pilot studies were then performed with a subset of 1,399 compounds in syndicate in different conditions. Each were defined as compound inducing a variation of the response higher than 3 times the standard deviation of the DMSO control that are represented by this purple area in this 1D screening panel. Hits are shown as blue circle, and our positive control, AZ1208, was regularly monitored and to assess the PIM3 stability, and is depicted as the yellow dots. You have here the Venn diagram summarizing the hit list from 2 pilot screen conditions and their shared hit list. We obtained a hit rate of 6.2% and 4.1% specifically for allosteric binders based on the comparison of the two conditions in absence and in presence of ATP. The last picture is an example of a dose response curve of a hit selected from the pilot screen. So the data were fitted using the tie binding fit equation from Genedata Screener. So data quality of primary screen was evaluated using different statistical parameters. First, we evaluated the assay quality using the strictly standardized mean difference, SSMD, across all plates. As you can see here, the SSMD values are consistently above 6, which is a strong indicator of the high assay quality. Next, we assessed plate-to-plate variability using the robust coefficient of variation of the fluorescence for both the neutral and the positive controls. The CV remains below 5% across all plates, confirming the low variability. And finally, we use the X-scan graphs to monitor the well homogeneity and detect potential compound aggregation. So here the blue curve shows a compound that induces PIM3 aggregation, which is an anomaly. And in contrast, the purple curve represents a normal scan pattern, indicating a uniform fluorescence distribution. So this scan helps us to identify and exclude problematic wells or compounds early in the analysis. Now that the statistics are validated, let's look at primary screen result. So we screen a total of 48,252 compounds from our diversity library. On the left, the scatter plot here shows the distribution of the hits, the non-binder, and the control across the screening plates. And from this, 1,866 binders were identified, giving a hit rate of 3.9%. So all screening data were processed using Genedata Screener, ensuring the robust statistical analysis. Compounds that induced fluorescence change greater than 20% were deprioritized, as they may reflect non-specific or artifacts. So out of this initial hit, 299 compounds were selected by our medicinal chemistry experts based on tractability criteria. These compounds underwent a hit confirmation assay, resulting in a strong confirmation rate of 72%, which supports the reliability of our primary skin. In the middle graph here, the line graphs represent the binding affinity curves for the selected compounds, confirming their interaction with PIM3. And on the right, we displayed the chemical structure of 6 representative PIM3 binders, which are now candidates for further optimization. So taking advantage that the previous study, HT-SpS study, using the same Eurofins discovery library was already performed, we conduct a comparative medchem study to evaluate chemical entities identified through the two distinct screening approaches, affinity-based and activity-based. And as shown by the Venn diagrams here, the overlap between the hits identified by both methods is limited, only 43 hits. This highlights the identification of new chemical identities that are not picked by traditional HT-SpS. And one final key point we'd like to highlight today is that the HT spectral shift screening enables the discovery of chemical matter which within previously unexplored chemical spaces. We compared the chemical spaces occupied by the hits from our earlier HT-SpS screening using ADP Glow, as shown here in green, the green dots, with those from the HT Spectral Shift screen shown in red, and using the Pfizer golden triangle representation. Both sets of hits largely fall within the oral drug-like space. And however, when we conduct a PCA, a principal component analysis, based on Morgan fingerprints, we observe that the HT Spectral Shift hits extend into additional distinct region of chemical space, as indicated by these purple circles. So this suggests the possibility of accessing new binding sites through these compounds. So to conclude, let me summarize the key messages from the PIM3 case study. HT Spectral Shifts enables the direct identification of binder and significantly improves both the efficiency and accuracy of drug discovery workflow. HT Spectral Shift is not only highly sensitive, it also allows for the rapid detection of small molecules and is flexible enough to be applied to other modalities, including fragments and peptides. And finally, our comparative studies clearly show that this innovative affinity-based strategy expands the accessible chemical space and drives novel hit discovery. So now I'll hand over to Vanessa to present our second case study on the molecular clues.
Thank you, Maud. So building on what we just seen, we wanted now to explore whether high-throughput spectral shift could also be applied to more complex interactions, such as protein-induced proximity. So WEE1 is a serine-threonine kinase that plays a key role in cell cycle regulation, making it as a promising target in cancer therapy. However, developing selective WEE1 inhibitor remains a challenge. So the idea here is to use a molecular glue concept to promote proximity-induced interaction, enabling selective degradation of WEE1 targeting other binding sites than ATP. So in this case study, we would like to screen and to find molecular glue against the WEE1, and we have already one well-described molecular glue compound in the literature that is able to bind WEE1 and the cerebellum and acting like a glue. So on the right side, you can see the cryo-M structure showing the ternary complex formation. As a proof of concept, we aim to identify novel molecular glue, and for this we screen 106 molecular glue and PROTAC against WEE1 kinase, and we screen against a well-known E3 ligase, the truncated cerebellum midi, that which retain the functional activity of the full-length cerebellum, but making it biophysics compatible. So here, based on this insight, we designed different screening conditions to help us to identify and distinguish simple binders and molecular glues. This slide gives us an overview of how we set up the three screening conditions. So on the first part, on the left side, we screen for binary interaction. So the idea here for the first screening condition is to find binder against the protein of interest. So here WEE1. The second screening condition is to screen binders, to screen against the cerebellum construct and to find binders with a binary KD value. And the third condition, in screening condition 3, we assess the ternary complex formation with a ternary KD value, and we calculate the cooperativity factor, which tells us how well a compound promotes the interaction between the two proteins. A cooperativity factor greater than 1 suggests a thermodynamically favorable ternary complex. So let's now look at the screening cascade we developed to identify We1 molecular glue. On the left, you can see the screening cascade we use to identify molecular glue and assess their degradation function. So after protein production and library selection, we developed a miniaturized ESA on 1536 wild plate format. A primary screen is performed and followed by a dose response curve that enables us to extract KD value for binary and ternary interaction. Hits when then profiled using various binding and activity platform available at Eurofins Discovery. And while protein degradation and proteomic study are ongoing and not shown today, let's now focus on the library selection. As you can see on this pie chart, it mainly includes a thalidomide-like focus set along with positive and negative control. On the right side, we have also included this literature-reported We1 molecular glue and PROTAC tested in the blinded way to see if we are able, at the end of this screening, to find also these We1 molecular glue degraders. So now let's take a look at the data generated for the primary screen. Here now it's an overview of the 3 screening conditions that we tested in parallel during the primary screen, all performed at single dose, followed by hit confirmation in dose response. As you can imagine, this generates a large volume of data and combination. That's why, to make sense of this, we use a Venn diagram as our representation of choice to classify binders and molecular glues. So in this diagram, all compounds within the blue cycle are cereblon binders, while in the magenta cycle, all are We1 binders. So this visual helps us quickly identify overlaps and unique binder across conditions. So let's now dissect the data in more detail to better understand the nature of these hits. First, let's have a look on cereblon and We1 binders with their binary KD value. So on this scatter plot, you can see on the x-axis the cereblon PKD value, and we identify potent cereblon binder in the nanomolar range. On the y-axis, we have the We1 PKD value, and again, we were able to identify nanomolar binders for We1. At the top right corner of the plot, we can clearly see compound with dual binding properties, with strong binding for cereblon and We1. So this is the literature reported PROTAC known to bind both targets at the same time. However, this representation doesn't allow us to identify We1 molecular glues, since by definition here the glue should not bind We1 alone, but only in the presence of cereblon. So to capture this behavior, we need a different representation. So let's move to the next slide. So on this scatter plot, the y-axis shows the ternary PKD value obtained under the screening condition number 3, while in the y-axis you have the cooperativity factor. So the dot in the bottom right here show compound that bind We1, and the dot size reflects their binary KD for We1. In the middle panel, we start to observe compound with nanomolar ternary KD value and high cooperativity factor, which are indicators of molecular glue behavior. We found the literature described glue acting this one that was reported in the literature. So it binds the cereblon with a KD of 15 nanomolar and show no binding to V1. But it formed a ternary complex with a KD about 339 nanomolar and a good cooperativity factor higher than 88. So interestingly, we also identify novel compound with higher and lower cooperativity. For those one at the top right, you can see that they are not reported as a V1 molecular glue, so we need further investigation to see how those compounds are working as a glue for V1. So now the next step is to profile these molecular glue hits across different binding and activity assays to better understand their mechanism. So we began by profiling the well-known V1 molecular glue across several platforms to validate their binding properties, but also to observe if they have any activity properties. So on the left, we have the kinase profiler platform. So we confirm that this compound shows no inhibition of V1 and appears to be selective across the broader kinase panel. We then run a kinome scan, which also shows no binding to V1. So that is consistent with what we observed during the screening. On the cereblon side, we confirm the KD value using different cereblon constructs, with and without DDP1. And finally, we reconstitute the ternary complex here. As expected, there is no binding of V1 alone, but upon adding cereblon in the well, we observe this complex formation, confirming that this compound is acting like a glue. The other molecular glue identified during the screen are currently being profiled as well using the same approach. So to conclude this case study using high-throughput spectral shift approach, we are able to identify cooperative, non-cooperative compound targeting V1 cereblon complex. So this cooperativity factor helps us to distinguish molecular glues from bifunctional compounds, and this represents a rational strategy for discovering and characterizing molecular glue. So of course, once a molecular glue is identified, especially in the context of targeted protein degradation, it's essential to perform degradation assay to confirm its functional properties. So to summarize, here are some tech messages that we would like to share with you. The high-throughput spectral shift platform is transforming hit-finding strategies at Eurofins discovery by combining direct biophysical screening and downstream analysis with Genedata. And as a CRO, we are proud to offer this cutting-edge technology as a service to support drug discovery projects. So if you want to know more about our case study or services, please reach us out via this email address. Thanks for the attention. Thank you, the organizer, for giving us the opportunity to share our latest advances in this exciting field. So we are now happy to move on to the Q&A session.